metabolism is all of the chemical reactions of a cell. Catabolism is the subset of those metabolic reactions that basically break big things to make ATP. And then the other side of metabolism is anabolic pathways. Anabolic pathways build things, and if breaking things releases energy, what do you think building things takes? Takes energy, right? So catabolic pathways produce energy, and in your textbook, whenever you see that little yellow explosion, that's always ATP, right? It's energy. So catabolic pathways break things down to make energy. Anabolic pathways use that energy to build things. And that's just the way that everything in the cell goes, right? And what happens is, especially because ATP does not last for very long, right? In a cell, ATP can exist as ATP for about 30 minutes, and then it falls apart into ADP, which is sort of like taking your dollar bill and ripping it into tiny pieces, right? It doesn't work anymore. It's not useful. So what you need to do is sort of couple these reactions, right? So when you have a catabolic reaction that's making ATP, you got to use that ATP to do something. And it may be that you're using that ATP directly to do some work, or it may be that, let's say you ate a giant cheeseburger and now you're going to spend the next 5 hours sitting around. That cheeseburger is going to be burned into energy, and then you're going to use that energy to build fat. Right, which is why, like, even if, let's say you didn't have a cheeseburger, right? Let's say you had fat-free gummy candies. You had a whole big, gigantic, little orange slices. Everybody eats those little orange slices that are coated in sugar, right? We used to love those when I was a kid. We used to beg my mom, please. So if you had like a pound bag of that, and you ate that, right? You would take that sugar, you would burn it to ATP, and because you're sitting around, your body would say, I don't need all of that ATP. I'm going to take that sugar, burn it, make ATP, and then build fat. And I'm going to send that fat to your backside, and there it's going to sit until you decide that you're going to move some more, right? So those reactions are always coupled. They're coupled in our cells, they're coupled in microbial cells, right? Microbes eat. If they have too much food, they might store that food as a polysaccharide granule. If they have even more food, they might store that in a lipid inclusion, right? Because they have to couple the food that they eat to some sort of storage molecule, because you can't leave ATP sitting around. So everything's coupled, right? So catabolic pathways, right, release ATP, and that is used to drive the anabolic pathways, right? So we release energy by breaking things, and we use that energy to build things. And of course, because we are organisms, whenever we see arrows, we think enzymes, right? Because whenever a chemical reaction occurs, it's always an enzyme that's doing that job. So, catabolic, anabolic, metabolic, those shouldn't be new phrases to you, and neither should enzyme, right? You should know enzymes. We call them biological catalysts, right? A catalyst is something that speeds up a chemical reaction. We are actually going to use some non-biological catalysts in this class. We're going to use zinc dust later on. And you're not going to remember it then, but I'm going to tell you just because, right, so you know that I'm not lying, right? You're going to use zinc dust to convert nitrate to nitrite. Because what we're trying to decide is, does an organism have an enzyme that can do that job, right? So a catalyst just means something that performs a chemical reaction. And metals, right, do that, and also enzymes do that. In the body, enzymes perform all of the chemical reactions that we are capable of performing, right? And we know from general biology that there's this very important spot on enzymes called an active site, and the active site is that place where the substrate is held onto, right? Sort of like your hands, right? You grab it, break it apart, let it go, right? So your hands are the active site there. So the active site is what performs this job. So we want to think about active sites, not just for chapter 5, but when we get to chapter 8, we're actually going to be talking about special kinds of enzymes in cells and talking about the active site, where their active site binds, and we're also going to talk about this other site that can be found on enzymes. Another word for other is allo. Like an allele is an alternate form of a gene, right? So that ALL for allele or allosteric, as you might have heard this in general biology, means another site, right? Steric means site. So active site is the place you're paying attention to. The allosteric site is another place on the enzyme where something can bind. And when we talk about this in general biology, right, so here's our normal reaction, right? So here's this enzyme binding a substrate, turning it into products, right? So since this substrate got broken into two pieces, we would call that a catabolic pathway, right? So we took something big and we broke it into two smaller things. That enzyme can be inhibited in a couple of ways. One way that it can be inhibited is that something can actually be in the active site, right? It's like if you have two little kids and one's already sitting on your lap, it's kind of hard for the second one to get on your lap, right? I have big kids now, so now it's dogs. I go to my mom's house, she has a pit bull, my sister has a pit bull, and they like to sit on laps because for some reason 70-pound pit bulls think that they're lap dogs, right? One's on my lap, the other one can't get on my lap, right? Because they're competing for that spot. So a competitive inhibitor competes for the active site. And this is not typically used in a biological system to any end, it just is something that happens, right? So certain poisons actually kill you because they compete for the active site of your enzymes. And they make it so that enzyme can't do its job. In fact, one of the things we're going to talk about today, the electron transport chain. There are poisons like cyanide, and the way that they kill you is they bind to the electron transport chain and keep you from making ATP and you die. So that's not typically a way that organisms, it's not something organisms use for function, but this other phenomenon here is. Now in general biology, we don't talk about that very much. We just talk about the fact that enzymes can be inhibited. And if you remember back, you did this lab, I think they did with catechol oxidase, right, which you get from potatoes as they turn brown. And you're trying to figure out whether you have a competitive or a non-competitive inhibitor, and so you add more stuff and see what happens, right? But what we want to focus on is the fact that this phenomenon here, this is kind of like an on-off switch for the enzyme. If something binds to it, it can shut off the enzyme, which is pretty handy. Especially when you are a bacterial cell who has the capability of eating tons and tons of things, but you're not always being fed those things, you can shut off those enzymes, kind of set them aside, right? Don't do your job right now, I don't need you, right? So this allosteric site, as it's called, because it's a site other than the active site, is a site of what's either called allosteric inhibition or non-competitive inhibition, and that allows that enzyme to be turned on and shut off, which we're going to be talking a lot about in chapter 8. We're going to talk about genetics in chapter 8, and one of the big things that we're going to talk about is how you can turn on and off enzymes, so you can turn on and off certain aspects of your metabolism. Yeah?
So the non-competitive inhibitor, so when it's in there, it's off?
Mm-hmm. So what it's trying to show you here is, so look at this shape here, how it perfectly conforms to the shape of the substrate, and then when this binds, it kind of changes it.
Distorts it.
Mm-hmm. Yeah.
Not all enzymes have the allosteric site, correct? Or do they all?
That's a good question. I don't know. I am not sure. I will say that not all enzymes are turned on and off using an allosteric site. Like one of the things we're going to talk about today is glycolysis. The enzymes involved in glycolysis are not allosterically inhibited, right? Because they're always on, because you always want to be doing that process. We're going to talk about some special stuff later on in chapter 8 where you actually do want to shut off some function of the cell. So I don't know. It could be that they all have a possibility of being inhibited, but not all of them are inhibited. That's the best answer I can give you without pretending like I know more than I do, which you don't want to do, right? Okay, so this is important twofold, right? It's important because we want to understand enzymes, because every time we think about what a bacterial cell is capable of doing, we think there's an arrow there, that means there's an enzyme there. And why is that important? Another general biology concept, that enzymes come from genes, right? Two guys got the Nobel Prize, their last names were Beadle and Tatum, and they came up with this really important thing that all of us today go, no doy. And they said that for every enzyme, there's a gene. Which seems simple to us, right? We know that. That's what you learn in general biology, right? You learn about protein synthesis, which is what? The transcription and translation of a gene into a protein. But this was monumental, right? Because what it says is, everything you do is genetic. Everything that you exhibit, and we would say in general biology, phenotypically, the color of your hair, the color of your skin, how tall you are, whether or not you have a genetic disorder, how your immune system functions, whether or not you're predisposed to cardiovascular disease or autism, it turns out, right? All of these things tell us something about your genetics. And that's important for everybody, but it's especially important for a microorganism because they're so small. You've seen them, right? They're teeny. We can't tell much about them from looking at them. So the way that we learn about microorganisms is to see what they eat. Because if we can see what they eat, then we can say, well, if you were able to eat lactose, then you must have lactase. And if you have lactase, then you must have the gene for lactase, and now I know one thing about your chromosome. And then I try to see if you can eat mannitol, or if you can reduce nitrate, or any of these other things that you're going to be doing in lab. And I start taking a tally. You can do this, you can do this, you can do this, you can do this. That means you have those 4 enzymes, that means you have those 4 genes, that means I know now more about your chromosome. And I started learning more and more and more and more and more about your chromosome, basically by looking at how you metabolize. And eventually, I will know so much about your chromosome that I can identify you. Not because I ever looked at your DNA, but because I looked at what you eat. And from that, I inferred what enzymes you have, and from that, I inferred what genes you have. And now if I know 3,000 of your genes and some other things about you, like the arrangement of your cells and the morphology of your cells and the Gram reaction and whether you form spores or whether you're acid fast, once I know all of those things, I can go, unknown bacterium, you are E. coli, which we couldn't tell just from looking at E. coli, right? When we looked under the microscope, we said single bacillus. That could be E. coli, but you're gonna see 2 other single bacilli today, and they're also gram negative, just like E. coli. They're not all the same, they just have those 3 things in common. So we can't tell a lot by looking at bacteria and other prokaryotes. What we have to do in microbiology, specifically bacteriology, is look at what they eat and backtrack that to this. And once we know enough about their chromosome, now we can say who they are. So this is the way that we identify microorganisms. So we're not gonna spend a bunch of time in class and lecture talking about this, but when we get to lab 11, we're gonna start learning things about bacteria, what they need to grow, what they like in order to grow, what they eat, and all of those things are gonna culminate in these unknown bacteria that you're gonna be issued. And you're gonna look at all these different aspects and identify them. And you're identifying them because you're looking at the way they grow, the way they metabolize, and you're inferring things about their genetics, right? Because we can't just look at them, because there's not much to see under a light microscope. We can see morphology, we can see whether they're acid fast, we can see whether they form spores, we can't even see their flagella, right? Today, you're not gonna see flagella. You're gonna put live microorganisms under the microscope and try to see them swim. And if they swim, you go, well, you must have flagella, because that's what bacteria use to swim. But you can't see them, they're too small. So this idea, this one gene, one enzyme, right, is very important, especially to microbiology, because it's sort of the foundation of how we identify microorganisms. And you might not think, well, so what? So you're gonna, I get to identify, you're gonna identify 1, 2, 3, 4, 5-ish, 5.2 organisms in this class. And you're like, yay. But when we're talking about the 0.1% of bacteria that cause disease, right, what's the first step in treating disease? Identifying it, right? So all of this stuff that you're learning how to do are the same things that people are doing in medical microbiology labs to identify pathogens, so that they can call the clinician and say, this person has E. coli, and that's what's making them sick. And then the clinician, right, and that's when you're done as a microbiologist. It's not like the clinician says, oh, what should I do? What should I do for them? I don't know, I'm a microbiologist, you're the physician. All I do is tell you what it is, and you treat. You give antibiotics, you give supportive care, right? You do all of the things to treat it. So the first step in any treatment is identification. If you don't know what's making a person sick, it's really hard to make them better. So even though it seems like, who cares, right? It's a really important thing, right? Because as we get into our disease unit, right, the first step in stopping disease is identifying what is causing disease. And once you've identified it, now at least you know who you're fighting, right? And the way that we do that is by all the things that we talk about in this chapter, right? Looking at what they can eat, because that tells us about their enzymes, which tells us about their genes, which tells us about their chromosomes, and allows us to identify that. So just kind of keep that in the back of your mind when you're thinking to yourself, I don't care why, right? Because everyone always wants to know why. That's why. Also because it's going to be on your test. And you want to get a good grade so that you can go to nursing school or PA school or dental school or wherever it is that you're going to end up. So that's why this is important, right? That's the nice thing about teaching a 200 level and rather than a 100 level. Because when my 107 students say why, and sometimes you're like, no, you probably never ever will talk about this again in your whole life. So it's like I tell my kids, it's because I said so, right? But at least you have this idea why, because you have the goal of treating people. And all of the things that you learn in this class are moving toward that goal. You may not personally do that, but somebody is doing all of this stuff behind the scenes to help you, right? Or even to tell you what precautions you should be using when you're working with patients. Why do they know that? Because there are epidemiologists and microbiologists that are working out those guidelines and giving them to clinicians. So that's why they make you take microbiology, right? Because it's important. Not just because I said so, but because it is, right? So one thing, though, that you don't talk about in terms of metabolism that we do want to talk about in this class is the fact that at the heart of all metabolism is this fancy thing called a redox reaction, right? So when I was in microbiology, my teacher said, life is one big redox reaction. That's how he began this lecture. And we were all like, what are you talking about? What is that? I don't even know what you're saying, right? So we'll break it down, right? Because that was a big time like Bio 376 or something like that, right? And this is Bio 205. So we're going to baby step into it. So redox is a sort of a combination word, right? It's like Brangelina. Is that what they call themselves, Brangelina, right? Brad Pitt and Angelina Jolie. It's like you took two words and you mashed them together. So redox is a mashing together of two words, reduction and oxidation. It's a mashup of those two words because one never happens without the other. And the reason for that is that these two words describe the removal of electrons and the gaining of electrons. And here's the deal with electrons, right? Electrons are charged. They have a negative charge. Whenever you have something that has a charge, it's unstable. It won't stay where you put it. Things with a charge want to go and get that charge neutralized. So whenever you take an electron from something, something's gonna take it, right? That's what happens when you get electrocuted, right? You touch a flow of electrons and they don't want to stay there, they want to move through your body, right? So electrons are very unstable, they want to move. So when you release them, they go somewhere, right? It's kind of like if you have a magnet and you pull it off the fridge, but you keep it close to the fridge, you let go, it'll go back to the fridge, right? Because it's got a charge, right? And it wants to have that charge satisfied. So whenever an electron is removed, it always goes somewhere. So in the first picture here, we have molecule A with that red electron. Now, whenever you see a little e, a lowercase e with a line in a textbook, that always means electron. It's a nice little abbreviation that you can use instead of writing out the word electron, right? So molecule A has an electron, and that electron is taken by molecule B in this process. And because there's an arrow in this process, what do we know is making that happen? An enzyme, right? So molecule A is said to be oxidized in this reaction. So the removal of an electron from a molecule is an oxidation reaction. But because that electron always goes somewhere, in this case molecule B, right? Then we have that coupled reduction, right? Because to receive an electron is to be reduced. So when we say redox, what we're saying is this oxidation and reduction that happened, right? Where did the electron come from and where did it go? And the reason why my teacher said that life is one big redox reaction is because this fuels the production of ATP. Pretty much all of the chemical reactions that you can think of involve redox. The removal of electrons that end up somewhere else, right? And that's where we're gonna focus our attention when we talk about metabolism. We're gonna follow the electrons. Because it turns out that when you eat, really the fundamental question we're asking about you is, where do you get your electrons? That's the question that's being asked. So either it was those gummy orange candies or the cheeseburger, maybe you ate some like lean chicken breast and some kale for lunch, right? Well, then that's where your electrons came from. Plants, what they eat is water, because that's where they get their electrons. So this question, where do you get your electrons? That is what you eat, right? So that's what we're gonna be looking at. Now, anyone here who ever thought maybe they wanted to be a chemistry major and then moved over into biology and took a bunch of chemistry classes may have heard that chemists don't like biologists because it's messy, right? Chemistry is much cleaner than biology. And even in redox, chemistry is cleaner than biology. So this one over here, right? I never know stage right. Is that stage right? I'm not sure. This one, that's sort of a chemical, clean reaction. This one over here, right, which is on your right or left, depending on which way you're facing, that is a messier biological redox reaction, and it involves a very important molecule that may look familiar to you, and that is NAD. And in general biology, you might have learned about the fact that NAD can become NADH, and you're like, what? So this here is a messier biological application of redox. And the reason it's messy is because in biological systems, right, because electrons are super crazy, they don't oftentimes move by themselves. They usually move as a hydrogen atom. Because hydrogen is a crazy atom that, unlike every other atom on the planet, doesn't have a neutron. Remember, you go back to your chemistry class that you all had to take to get into this class, right? When the subatomic particles, electrons, protons, neutrons. But if you look at a hydrogen atom, it has a single proton surrounded by a single electron. So in biological systems, we watch hydrogen atoms move. And when the hydrogen atoms move, we go, oh, well, that's where the electrons went too. Which is nice because in most biological reactions that you look at, all you have to do is say, that one has no H's, that one has H's. So the electrons went from him that has no, right, to this guy. And now he's got the H's, so he's been reduced, right? So watch the H's. So here we have some organic molecule that has two hydrogen atoms on it, right? We don't know anything else about it, we just know it has some H's. And through this process, this redox process, this molecule here loses those hydrogens. He becomes oxidized. NAD is what collects those electrons, and so he becomes NADH, right? So the purple guy, NAD, is reduced. So NAD and NADH are the same thing. NAD is its oxidized form, NADH is its reduced form, but it's the same molecule, right? It's like, this is Krista with glasses, it's still Krista, right? It's just a little extra. So NADH gets converted to NADH and goes back and forth and back and forth and back and forth, and that's why NADH is such a cool molecule, because it can run around the cell, grab electrons, and take them somewhere else, right? So this is messier, right? So this organic molecule that includes hydrogen atoms, that's what you're eating. That's your cheeseburger, or your kale, or your chicken, or your tofu, right? Whatever your poison, frozen yogurt, I don't know, right? Whatever that was, it had hydrogens on it when you ate it. And you broke it into smaller hydrogen-containing particles in your digestive tract. And then you moved those small hydrogen-containing molecules out into your bloodstream. And then they went into your liver, and your liver broke them into really small pieces and sent them back out into the blood. And then they went into cells, and ultimately inside the mitochondria of your cells, you pulled all of those electrons off. You ate them, right? What we're going to talk about today is why this creates ATP, right? How is it that I can take something and just by oxidizing it, get energy from it, right? Now it turns out, right, if you think about it, this is not microbiology, but every time that you have a bonfire, you're doing this kind of thing. You're taking wood, which has tons of H's, you're lighting it on fire, and you do that, right, because it's nice and the ambiance is nice when you have a fire, but it's also warm, right? So when you take that big chunk of wood and you oxidize it to ash, you get energy from that. You get light energy and you get heat energy, right? And that's, by the way, a byproduct of this in your body is your body temperature. The reason you can maintain a body temperature of 98.6 degrees is because when you oxidize your food, you give off heat. You get ATP, but you also get heat. So we're going to be talking about, right, first, how this makes ATP. Why is it that when I say I'm eating, what I'm saying is I'm taking electrons from this thing, why does that make energy for me? Are you ready? The reason that this right here, right, taking electrons from something, oxidizing your food, right, eating food is oxidizing food. Burning wood to ash is oxidizing wood. Taking gasoline and putting it in your car is oxidizing that fuel, right? All of those processes take some fuel and burn them into energy, right? So why does this make energy for you? Because this process of oxidizing food is coupled to the reduction of ADP. Right? So here we have the electron donor. Right? Somebody who has a bunch of electrons. So here's your cheeseburger. You took all the electrons off the cheeseburger, gave them to NADH. NADH comes over here and he gives the electrons to this molecule here, which has 2 phosphate groups. That's ADP. And this little guy over here. And when he gives those electrons to ADP, ADP becomes ATP. That's how eating electrons gives you food. And you're like, really? Just an arrow, right? Don't you worry, I'm gonna show you what that arrow is, right? But that's why we talk about eating electrons, right? That's why life is one big redox reaction, because you take something, your food, you oxidize it, you use those electrons ultimately to reduce ADP to ATP, and that's your energy. And you use that energy to do whatever work you want to do, right? So that's why life is one redox reaction. So not one, lots of redox reactions, right? So what we're going to be talking about today is basically this phenomenon here, making ATP. And then we're going to sort of piggyback that concept and see different ways that ATP can be used to fuel other processes, like photosynthesis and chemosynthesis, which is a new one probably for you, right? So you eat electrons, you give those electrons basically to NAD, which becomes NADH, and NADH uses those electrons to make ATP, right? And that's what we're going to talk about today, is how that process happens. And we're going to do that in the context of 2 things that you've already studied, right? So here's the chemical formula for respiration. Now I want you to think H's, right? So here is glucose, C6H12O6. Would you say that that has a lot of H's? I would say that has a lot of H's. Notice that glucose, and this is no surprise to you, you know that glycolysis and the Krebs cycle and the electron transport chain start with glucose and end with CO2. Here's your glucose, here's your CO2. That's not new stuff. But what we're doing now is putting on the lens of redox and saying, oh, well, there were a bunch of H's and now they're gone. So that thing has been oxidized. And you go, that I get, right? So somehow those electrons are used to make ATP, and that's true, right? The other part of this process is that you have to breathe, right? The whole vital force thing that it turns out actually was a thing, even though people mocked them when it was really a thing. It's a thing, right? It's oxygen. And oxygen through this process, right, because oxidations are always coupled to reductions, you can't have respiration without oxygen because somebody has to collect the electrons. And oxygen is very good at collecting electrons. It loves electrons. It's like the super most negative person in the whole world, right? So this process of respiration, the conversion, right, the oxidation of glucose to carbon dioxide, right? So that same cheeseburger, tofu, chicken, kale, whatever it was you were eating, guess how you get rid of it? You exhale it. Isn't that weird? I mean, you poop a lot of it too, right? But I'm talking about the part that actually gets into your body. The part of the cheeseburger that gets into your body is exhaled as CO2. Which took a long time for people to figure out, right? Because if I just asked you, you breathe in oxygen, you breathe out carbon dioxide, what I would assume, and probably what most people would assume if I just thought, was I breathe it in, it gets converted to CO2, and then I exhale CO2, but that's actually not the case at all. Your food becomes CO2, your oxygen becomes part of your urine. Isn't that weird? That's chemistry, baby, right? So this process of respiration, whose main goal, right? So respiration includes glycolysis, Krebs cycle, electron transport chain. The main goal of respiration is to make energy, ATP, right? So up to 38 ATP per molecule of glucose. I'm not going to ask you that, don't worry about it, right? But lots of ATP. Now, this process, it turns out, can happen in the absence of oxygen. And you're like, oh, my general biology teacher told me if we're not in the presence of oxygen, then it's fermentation. No offense to your general biology teacher. If they said that to you, they were wrong. If a website says that to you, they are wrong. It's not whether or not there's oxygen there. This, then, is not the only kind of respiration, but it's typically the only kind of respiration you talk about in general biology, right? So we just call it respiration, which is accurate, but not precise. There's a difference between accuracy and precise. Like accurate is I weigh between 100 and 200 pounds. It's not super helpful, right? Like if you were going to pick up someone from the airport and you'd never met them before and someone said, well, they're between 10 and 77 years old. That would be accurate if you were talking about me. It really wouldn't be helpful, right? So precision means something that is more close to the truth. So something can be accurate and not precise. So this is accurate, this is respiration, but it is not precise because it doesn't tell you what kind of respiration this is. And this respiration is called aerobic respiration. Because any process that requires oxygen is called aerobic. And this requires oxygen. So in this class, we have to be more precise. We can't just say respiration. We have to say that that thing that you learned in general biology, which is done by all eukaryotes, by the way, plants, fungi, protozoans, who am I missing? Animals, you and I, we all do this, which is why they talk about it in general biology, because that's what all the big guys do, right? But today, we're in a microbiology class, and in a microbiology class, we want to focus not just on those guys, but on the little guys, right? And it turns out the little guys have other ways of undergoing respiration. So we have to call this aerobic respiration, right? So respiration is what we're going to talk about today, all the different ways that respiration happens and all that fun stuff, right? On Tuesday, we're going to revisit this concept of photosynthesis that you talked about in general biology, right? So photosynthesis is this process right here, right? Energy in the form of sunlight and carbon dioxide, which notice has no H's, and water, which has H's, produces a carbohydrate. There's another nice abbreviation for you. Whenever you see CH2O, that just means a sugar, right? And then in a lot of textbooks, there'll just be a number here, or N, right? Because carbohydrates are just little chunks of sugar all stuck together. So this process yields a carbohydrate, water, and oxygen. So what happens, right? When we look at that process in more detail, the CO2, right? It's just the opposite of what happened up here. So the CO2 gets reduced to a sugar. So the plant breathes in CO2 through its cute little stomata. They look like little lips on the bottom side of a plant leaf. It gets reduced because the water that they take up in their roots provides the electrons to reduce it. So they oxidize water in order to reduce carbon dioxide. This is what they want out of that process, because that's the food. This, that we love them for, is just a byproduct of that. It's garbage. They just get rid of it. Notice this, CH2O. If I put a little 6 right there, then it would be C6H12O6. So they're making a reduced molecule that they will build into a larger reduced molecule, and then they will send that to their mitochondria, where they will undergo aerobic respiration and make ATP, which sounds super stupid, right? Like, if you can make ATP from the sun, why do you have a mitochondria? And I don't know the answer to that because I did not design plants, right? But they go through all this process of making their own food so they can eat their own food, right? So this process here that you learned about in general biology, right? So this right here is the light and the dark, or the light and the light independent reactions of photosynthesis, the whole kit and caboodle, right? We start with light, carbon dioxide, and water, and we end up with some kind of sugar. And we call that photosynthesis, because the synthesis of the sugar is driven by light, right? So like protein synthesis makes proteins, right? This is photosynthesis. Now, those of you who are nitpicky might have noticed that this is off center, right? For the same reason that's off center, this is not the only kind of photosynthesis that there is. Why is this the only photosynthesis you talk about in general biology? Because this is what plants do, and this is what protists do. So all of the eukaryotes on the planet, they all do this kind of photosynthesis, but it's not the only kind. So we're now, instead of just being accurate, we're going to be more precise, and we're going to call this oxygenic photosynthesis. So genic means to create. Oxy means to generate oxygen. And if you look at this process, you had water, and what did you oxidize that water into? Oxygen. So this is oxygenic. Now, in the beginning microbiological student's mind, aerobic and oxygenic can get tangled up, right? They're the opposite. Aerobic needs oxygen. Oxygenic creates oxygen. So make sure you keep those straight. Just think genic, genesis, right? Making. So on Tuesday, we're going to revisit oxygenic photosynthesis, because there are also bacteria that undergo oxygenic photosynthesis. But just like this aerobic respiration is going to get added to, oxygenic photosynthesis is going to get added to. We're going to talk about anaerobic respiration, and we're going to talk about anoxygenic photosynthesis. So aerobic means needs oxygen. What do you think anaerobic means? It doesn't need oxygen. Oxygenic means generates oxygen. What do you think anoxygenic means? Not generating oxygen, right? Because an or a always means not to whatever it's attached to. So anaerobic, not aerobic. Anoxygenic, not oxygenic. So this is today, and this is next Tuesday. What we're gonna focus on first though is the energy that it takes for all of this stuff to occur, and that is energy in the form of a beautiful little molecule called adenosine triphosphate, or ATP. So remember I said there are two ways to make ATP. Two ways. Here's way number one. Way number one is called substrate level phosphorylation. So phosphorylation, right? That, okay, right? Phosphorylate means to add a phosphate group to. And what's the difference between ADP and ATP? ADP is adenosine diphosphate. ATP is adenosine triphosphate. So what have you done to that molecule? You've added a phosphate, which means you phosphorylated it. Substrate level phosphorylation is a phrase that describes where the phosphate came from. And it just came from a substrate. So some molecule actually just hands a phosphate group to ATP. So this, I know you all thought when you got done with general biology, like, thank God, never, ever, ever again, right? Here is one half, the second half of glycolysis. Now, what I would like to point out to you is not all the fun words, although if you learn how to say them, you sound smart to people, so that's cool, right? I want to focus on these arrows, right? What do the arrows mean? There's an enzyme involved in that process, right? So glycolysis, here's why we care about glycolysis, fundamentally. Just what its name says, it splits glucose. That is the main job of glycolysis, right? The main job of glycolysis is to break glucose, right? So that's step one, glycolysis, right? Why? Because you break glucose. It's sort of like taking your steak and cutting it into bite-sized pieces. Just makes it easier to eat, right? So you take glucose and you break it into this thing, pyruvate. So here we're starting in the middle, right? But here's pyruvate at the bottom, right? How many carbons does glucose have? 6, right? Which gets converted to a 3-carbon molecule. So all you've really done is taken glucose and broken it apart, which is good because the Krebs cycle, which comes next, can't eat a glucose molecule. It needs it cut up into little bites for it, right? But what we're focusing on is sort of a fringe benefit of glycolysis. And a fringe benefit of glycolysis is that a little bit of ATP is made. Made here and made here. We're going to zoom in. Notice I haven't said the names of any of those, and it was intentional because I don't want you to focus on the names of the enzymes, the names of the products, the names of the substrate. I don't care. Glucose, pyruvate. But we are going to zoom in right here to this yellow arrow, and the enzyme represented by that yellow arrow is over there, and his name is pyruvate kinase. I don't even really care that you know his name, but we're going to use him as an example of substrate level phosphorylation. So if you look at the substrate for this enzyme and the product, notice the substrate has a phosphate group and the product does not. So what did that kinase do? It removed a phosphate group. So it's very common for enzymes to remove phosphates to be called kinases, right? So if you hear that, like creatine kinase, that's important to bodybuilders, right? Because creatine is sort of an intermediary for ATP, right? But this has a phosphate, this does not. That enzyme takes the phosphate from this molecule, phosphoenol pyruvic acid, and gives it to ADP. So it takes into its active site phosphoenol pyruvic acid and ADP. It takes a phosphate group off, plows it into ADP, and you get ATP and pyruvate. The important part of this process is the pyruvate, because that's what goes on, right? Because pyruvate is what goes into the Krebs cycle. This is just a fringe benefit, get a little ATP, right? But that is a great example of substrate level phosphorylation. Substrate level phosphorylation simply means I take a phosphate group from you and I hand it to ADP directly. No middleman. That is one way of making ATP, and it is not the biggest way of making ATP, but it is a way of making ATP, right? So we're gonna keep that in mind. Yeah, that's great, glycolysis, you do that for us, but what we really care about, right, is the fact that you're making pyruvate. Why is glycolysis necessary? To make pyruvate, right? To cut the steak into pieces so that you can put them in your mouth and eat them. The big way of making ATP is not substrate level phosphorylation, it's something called oxidative phosphorylation. Oxidative doesn't mean oxygen. Oxidative means redox. Because what is the oxidation? What is the definition of oxidation? What are we removing? Electrons. So oxidative phosphorylation means phosphorylation as a result of electrons moving. And it happens, notice I've totally skipped the middle part of respiration, because you've already done that, right? We gotta build, we gotta get bigger, we gotta get deeper, right? So we said, oh, glycolysis, you're so cool because you make pyruvate for us, and we haven't talked at all about the Krebs cycle. I'm gonna tell you a little secret, we're not gonna talk a lot about the Krebs cycle. Because it's a bunch of enzymes, which are super cool, tells us lots of things about the chromosome of an organism, but because they all have it, it really doesn't tell us anything unique about them. It's like saying every person on the planet has a nose. So when I describe people, I'm gonna say, okay, so she's 5'3", she has a nose. You're like, thanks, that's not really helpful, right? So we're not gonna study all the enzymes of the Krebs cycle because everybody's got them. It doesn't really tell us anything important about the cell. So what we're gonna do instead is say, what does the Krebs cycle do that's important for the cell, right? And what the Krebs cycle does is takes this guy right here and converts that 3 into a 1 carbon molecule, which is CO2, which is garbage. So that's not the important part, because that's the garbage, right? But what it's doing is something that's hidden when we only follow the carbons, which is why we miss it a lot of times when we study general biology, because in general biology you're talking about the 6 carbon becoming a 3 carbon becoming a 1 carbon. What we missed was that this molecule had 3 H's, and this one has 0 H's. 3 H's means electrons. 0 H's means no electrons, but what do we know whenever there's an oxidation? What does there also have to be? A reduction. Somebody's got to collect those electrons, right? And in comes that cute little guy, NAD, who takes these electrons and becomes NADH. So that is the only thing that we care about, Krebs cycle, because look right here. There's your NADH. So what is the Krebs cycle good for? Collecting electrons. Scoops them all up and brings them to this magical place called the electron transport chain, whose name is very descriptive, unlike lots of things in biology. It is a chain of electron transporting proteins. But here's the problem with naming it the electron transport chain. It's too obvious. Because if we watch oxidative phosphorylation happen, right? Just these guys. I say, okay, oxidative phosphorylation, right? Okay, so I'm watching electrons move. I see electrons move. I don't see any ATP here. So when I talk about the electron transport chain, where's the ATP, right? That's the whole point of the electron transport chain, is to make ATP. Where's the ATP, right? So calling it the electron transport chain almost does a disservice to the process. Really what they should have called it is the proton pump, because that's what it's actually doing, right? There used to be this show on PBS called The Verbivores, and before the internet, you could call them and be like, what does this word mean? And they were like Google, right? And they knew this stuff off the top of their heads. And I used to teach a Saturday class, right? Because that's what happens when you first start teaching. You got to teach at 9 and teach Saturdays, and it's lame. It's lame for you too as a student, right? But we do it because sometimes you got a full-time job. You got it. So I'm driving, and someone calls up and they said, I want to know where the word prestidigitarianism came from. And I'm like, oh, that's interesting. What is that word, right? So prestidigitarianism is the technical name for what magicians call sleight of hand. And the idea is, right, sleight of hand, the magician distracts you and then does this little thing, right? So you're paying attention to the big thing and something else is happening, and then all of a sudden your wallet's gone and you don't, your watch is not on your wrist anymore, right? Because you've been paying attention to something else. I think that naming this the electron transport chain is kind of a sleight of hand because it makes you think, right, if it's an electron transport chain, it must be the electrons that are important. But here's what happens to the electron. You do all this work, you break it apart, the Krebs cycle breaks it apart further, you bring the electron over here, it goes through here, and you urinate it out. So obviously it's not the important thing, or you wouldn't be peeing it at the end of this process. Because guess what, it's done, right? You urinate it, it's gone. Right, you don't drink your urine, it's waste. Right, even if you're dehydrated and you're dying, you don't drink your urine because it's waste, right? So that's garbage. So what's really important about the electron transport chain is that when the electrons move, they pump protons. So notice this process. The electron comes in and it moves through and it becomes garbage. But while it's doing that, look at what's happening to these protons. They're going from this side of a semi-permeable membrane, which by the way is not permeable to ions. Even though protons are tiny, they're charged. So they can't get through, they have to be pumped, right? And pumped because they're gonna be in higher concentration here. So we call that the translocation of protons. Trans, like transcontinental railroad, across the continent. Translocation, across the membrane. So protons go here, protons go here, protons go here. That is why you have to have an electron transport chain. Because if you don't have an electron transport chain, you can't move protons. Yeah?
So, then what would you call it? Is it the oxidative phosphorylation?
So hold tight, we're not done yet. So this, right, this is just part one of the parade. And that's the other thing about saying, oh, electron transport chain, right, we're not done. We've finished with the electrons, we've moved the protons, we have no ATP. So this whole process really isn't the last step in the parade, it's the third of 4 steps in the parade, right? So the electrons are on food. Food gets broken into 2 pieces in the glycolysis, gets broken into just electrons in the Krebs cycle. Electrons go into the electron transport chain, they move through it and get discarded as waste. Protons move across, and that's not the end of the story. The end of the story is this little cutie right here. And his name is ATP synthase, or in some textbooks, ATPase. So enzymes, right, a lot of times we recognize enzymes because they have the name ACE at the end of them, right, like pyruvate kinase, or ATPase, or ATP synthase, and in this case, the name is a lot more descriptive than pyruvate kinase. ATP synthase makes ATP. And the way that it does that is by allowing protons to move through it. When protons move through an ATPase, right? So an ATPase is a channel protein, right? You learn about those in general biology. A channel protein is something, it's like a door that allows things that normally wouldn't be able to get through a plasma membrane to get through a plasma membrane, like ions. Ions can't get through a plasma membrane. But if you've got a hole in the plasma membrane, a channel, they can flow through. But this is a very special kind of channel protein, right? It has a channel, but it also has this enzyme attached to the channel. And that enzyme, as protons flow through it, it causes the enzyme to move an ADP and a phosphate group closer together. So you can imagine, right, if this is my ADP and this is my phosphate group, right? I am the enzyme part of ATP synthase. One proton flows through, right? Because that positive charge is charged, it makes me move, right? Because I've got negative, and I go like this. And then another one rolls through and I go like this. And another one comes through and I go like this. And I make ATP. So this whole process, there's the phosphorylation. Oxidative refers to all of this. Because each of these is a redox, right? Each of those, when NADH comes and donates the electron to him, he becomes oxidized and he becomes reduced. When he donates the electron to him, he becomes oxidized, he becomes reduced. So it's oxidative. Oxidative just describes this sort of flow of oxidation, right? Because oxidize, 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 and finally, right, everybody's oxidized and we're done. So it's trying to tie those two things together, oxidative with phosphorylation. So notice there's no molecule giving a phosphate group to ADP, right? This protein, this enzyme here is actually building ATP from ADP and a free phosphate group. So whereas in the other example, we had a substrate donating a phosphate group, here, these two molecules are just sitting in that ATPase and then they get joined together. So oxidative phosphorylation encompasses this whole thing. The oxidative portion describes all of the electrons moving through, and the phosphorylation describes what happens here as that ATP synthase sort of ratchets together and makes ATP. Yeah.
What two things does the enzyme push together? Like you just said it.
ADP and phosphate. So we call that oxidative phosphorylation, not substrate level phosphorylation, because oxidative phosphorylation relies on an electron transport chain. So the magic is in those teeny tiny protons that we didn't pay attention to. The magic is in those protons that are slowly building up, right? Because imagine you take these protons and you put them all here, and you just keep doing that over and over and over again, right? So you've got this mass of protons. And protons don't like being crowded for two reasons. Number one, nobody likes to be crowded, right? So what do molecules that are crowded together like to do? Diffuse, right? They want to move away from one another. And that's a totally passive process. Molecules will do that without any input of energy, right? Do you ever do that? You know, sometimes they do experiments, right? Where you take a bottle or a glass of water and you just put one drop of food coloring in there. You don't touch it, don't look at it later on, right? The whole glass is going to be colored because those dye molecules will diffuse out. So molecules don't like to be crowded. So as we push more and more proton up here, they get crowded and they really don't like it because they all have a positive charge. Right? What happens when two positive charges, right? They're constantly banging apart from one another because they don't like being next to one another. That creates, right, and this is a word that's used for more than just this, they call it a proton motive force. There's this huge amount of protons stuck together. All they want to do desperately is get away from one another, and the only way they can do that is to go through that channel. And when they go through the channel, you exploit that. You use that energy, right? It's sort of like a water wheel, right? Or like a hydroelectric dam, right? So you have water that wants to run downhill because of gravity. You don't have to make it run downhill, and it will run downhill, and that'll be just fine. But you can put a paddle in that water, and the water running down the hill will spin your paddle, and you can use that to generate electricity. You can use that, right, in not really ancient times, but they used to use that to grind grain. And you're just exploiting that natural movement of water. Here, you're exploiting the natural movement of the protons. The protons are crowded, and they're all positively charged, so they want desperately to get away from one another, and the only way to do it is through here. And then you get ATP. And that's how you survive. So this ATP, right, lots and lots of ATP, generated in the mitochondria of eukaryotes, generated at the plasma membrane of bacteria, because bacteria don't have membrane-bound organelles, and this absolutely has to happen in a membrane. So like there are poisons that'll kill you, and they can either bind here and stop this, or some of them do, they make it leaky. So So now the protons just fall through and you die because you're eating, you're breathing, everything's going right, but all of those protons are just going here instead of going through here, and you die because you don't make any ATP. So how do you, every organism on the planet make a lot of ATP through oxidative phosphorylation, right? When they say people, it's like 32, 34, something like that, right? High 30s ATP. You make less ATP than they do because you have to take things and put them in the mitochondria. So bacteria can usually make 38 ATP per molecule of glucose. Now, I said we don't care about the Krebs cycle, but I do want to say that there are some organisms who don't have a Krebs cycle. They don't have the genes, so they can't make the enzymes, so they can't do this. So they actually never undergo oxidative phosphorylation. They're left only with that substrate level phosphorylation that we talked about earlier, which means they're very slow growing. Because whereas E. coli can make 38 ATP for every cheeseburger he eats, these guys can only make 2 ATP for every cheeseburger they eat. If you can only make 2 ATP, you can't grow that fast. So there are organisms who don't do Krebs cycle or electron transport chain. But every organism that has the enzymes, right, has the Krebs cycle enzymes. That's the common pathway for everybody. So if you do this, you've also done Krebs cycle. And Krebs cycle for us is important because it oxidizes your food completely so that you have all of these electrons, right? And why is it important to have lots of electrons here? Because every electron is going to move a proton, and every proton that goes back through an ATPase is going to be ATP that you generated, right? So the more H's in what you eat, the more electrons you have available for the electron transport chain, the more protons you'll move, the more ATP you'll generate. So by far, right, the most efficient way of generating ATP is through the electron transport chain, right? And really the only difference, right? So everything that can do this, plants, protozoans, protospect, all of those that can undergo respiration, they all have this, right? The difference between you and a sulfur-eating bacterium or an iron-eating bacterium or any other organism is just this. Who gave the electron to NADH? So this is it's a common pathway for everybody. The difference is, where'd you get your electron, and what do you do with it when you're done? Who gave it to you, and who takes it at the end? So this picture here is a picture that works for people and all animals and all fungi and all plants and lots of bacteria, right? Oxygen sits at the end of that electron transport chain, right? So this whole process is the same, oxidative phosphorylation. Difference is where did the electron come from and how do you get rid of it, right? And why do you have to get rid of the electron? Let's do a little experiment. Are you ready? Let's do something fun. All right, let's say that I am NADH and you four are these guys. And Nassim, is that right? Nassim is going to play the role of oxygen. Are you ready? All right, so I'm an NADH. I'm going to come over and I'm going to give you an electron. And now I'm oxidized, right? So you're here and you're like, I really didn't want to be negative, Krista, right? So how can you alleviate that negative charge? You can pick up a proton. But it turns out that sitting right next to you is somebody who likes negative or electrons even more than you do. So you have your electron and Liz snatches it from you. Snatch it so we can have a demonstration. You weren't even touching it, Brent. So now, Brent, you're like, oh, that's so sweet. Brent doesn't like being negative, but Brent doesn't like being positive either because being positive is kind of an irritating thing, right? Like, I listen to this podcast and I really like it, except one of the girls is, oh, so he doesn't like being positive either, right? So what can you do if you don't want to be positive? You can get rid of the proton, right? But here's the magic, right? He picked it up here, he drops it off here. Now Liz, even though she likes protons or electrons, doesn't like to be negative, so what can Liz do? Pick up a proton. And then we have Katie. Katie likes electrons even more than Liz does, so what can Katie do? If you like electrons more and it's survival of the fittest, you just take it, right? It's like playground rules. If the yard duty teacher doesn't see, it didn't happen. She also does not like being positive, so she deposits her proton over here, and then Liz, and forgive me, Saluma. Dang, that was so close. You like electrons even more than baby does. And so what can you do if you're bigger, badder, and you like it more? You take it. Which is the hardest part about this demonstration, because no one ever likes to take things from people. Either you don't know them or they're your friend, right? You feel awkward, right? So now we have someone here who didn't like being negative, also doesn't like being positive, so she deposits her proton, right? That's our pump. Now we have Nassim, who is oxygen. Nobody on the planet likes electrons as much as oxygen. So you steal it, right? And you put a cap on it, and now it's water. Now you're no longer oxygen, and so Nassim goes away, right? So pretend Nassim is not here. We're not going to make her leave the room because then she wouldn't be able to be part of this demonstration. But I, NADH, am going to come over again, and I'm going to drop off an electron and steal, right? Let's make this happen. Come on, people. Move, move. Protons, 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 protons. Oh, no, you're not here anymore. She can't steal it. She's not here anymore. She became water. No offense, Nassim, we urinated you out, right? So NADH comes over and I go, hi, here's an electron. And it gets stolen, stolen, stolen. Protons are moving. Oh, now you can't take an electron because you already have one. And I come back over and I'm like, oh my God, right? And I'm so happy to get rid of it because it's a negative thing. And up, it stops. And then I come over and I bring another electron and I hand it to you, and you can't send it anywhere, but I am still doing my job because I'm a good electron carrier, and I come here and I can't give it to you. So without the electron acceptor, this comes to a grinding halt, right? So you don't just need to eat, you need to breathe. Either of those two things being gone, and this process shuts down, and if the process shuts down, no ATP. So everybody eats electrons, and everybody uses them to make ATP, which means that we could take and look at everybody on the whole planet. All right, this is crazy, right? This is a chart in which we can divide every single organism on the planet. We first divide them into organisms who eat and organisms who make their own food, right? So today, we're talking about the heterotrophs. And we talked about their electron source, right? What's your food? What electrons are you eating, right? And it turns out, right, that they all like organic electrons, like cheeseburgers. Today, we are talking about a subset of those organisms who have a chemical energy source and who are undergoing respiration. But notice, respiration, aerobic respiration that you talk about in general biology, look at what a small piece of the pie that actually is. You talk about this in general biology, and you talk about this in general biology. Because this and this are what eukaryotes do. I'm not going to show you this. I don't want you to freak out and think you have to memorize this. What I want you to understand is we're getting a bigger picture now, right? We're looking at more things. So this is general biology. This is general biology. Today, we're going to add to that. We're going to add to fermentation by talking about these things down here, and we're going to add to respiration by talking about this thing over here. And then on Tuesday, we're going to switch to these guys, right? And you talked about this in general biology, and we're going to build on that. We're going to talk about oxygenic photosynthesis and anoxygenic photosynthesis. I gave it a pretty little gradient because purple and green bacteria undergo anoxygenic photosynthesis, so I thought it was pretty. We're also going to talk about organisms who make their own food not using sunlight but using chemicals, and we call them sulfur oxidizers and iron oxidizers. So today we're going to build onto respiration and fermentation. Tuesday we're going to build onto autotrophy, onto photosynthesis, right? So we just talked in depth about aerobic respiration. We didn't go through all the enzymes and all the substrates like maybe you did in general biology, because I know sometimes, just to throw something at you, they'll be like, memorize these 3 enzymes or something like that, right? We don't care. Why is glycolysis important, right? Because it cuts the steak up, right? Why is Krebs cycle important? Because it takes all the electrons off the steak so they can go to the electron transport chain. Why is it important? Because it's translocating protons. Why is that important? Because they're flowing through an ATPase, and that is respiration. So, when we talk about heterotrophy, we can ask a couple fundamental questions. Number 1, what kind of carbon, right? What are you eating? And it's always food. It's very simple. The answer is always the same. It's always organic. We're always eating things, right? Where do you get your electrons? Sugar, which comes from your food. What do you do with them? I throw them away. Ultimately, the product of your food is waste, right? Either stool, because it's moved through your digestive tract unabsorbed, or as water and carbon dioxide, because metabolically it was waste, right? So what does that mean for this? This molecule right here is your carbon, is your electrons, is your energy. Everything that you need comes from this guy right here. So that means sometimes in your life, you will not break him completely to CO2, you will build him into something else, right? Like anybody who eats a fat-free diet can still get fat. Anybody who eats a sugar-free diet can still have blood sugar, because you can take those molecules and break them apart and build them into whatever you need, because you have amazing magical enzymes that will do that work for you, right? So this process, right, cellular respiration, so this is a zoom in of what you learn in general biology, right, is just a piece of the puzzle. General biology says to you respiration uses oxygen, which is true, but not all respiration uses oxygen, right? But this is kind of nice because it allows us to say, why is glycolysis? Oh, right, because it breaks it apart. Why is the Krebs cycle? Oh, right, because it makes NADH. Why is this important? Oh, right, because protons, right? So it gives us kind of a general overview, but it's not precise to say that respiration uses oxygen. It is accurate. You could say respiration uses oxygen. The precise way of describing respiration is to say respiration uses inorganic electron acceptors, like oxygen. Now, a cheater way of figuring out if something is organic or inorganic is to look for H's. If there are no H's, it's probably inorganic. Isn't that nice? Like C6H12O6 has H's, it's organic. CO2, no H's, inorganic. So here, I know everybody always loves pictures, especially when they're hard to figure out, right? So like, what am I looking at? Here, you are just looking at that electron transport chain, kind of oriented just like we did right here, right? So this thing that says FP, that's you. And this thing that says FES, that's you. And this site B is you. And the site O is over here, and you're the oxygen. This is then aerobic respiration, because it requires oxygen, right? Nassim was our oxygen molecule. If there's no oxygen present, we already learned what happens. Everything grinds to a screeching halt and you die, right? Not if you're a bacteria. If you are a person and you don't have oxygen, you will die. But bacteria, notice FP, FP, FES, FES, site B, site O, nitrate reductase. So E. coli and lots of other bacteria, right, when they don't have oxygen, they can go into their genome and transcribe and translate a gene into this enzyme, and this enzyme will take nitrate and have it collect the electron and become nitrite. So basically what that means is, if you're robbed of oxygen, right, we have no more oxygen, that means that you could come in as nitrate and you could collect the electron. And then you could take hers, and you could take hers, and you could take hers, and then I could bring another electron in, right? So let everybody do, so they can see you. Do it, no, that way, right? and then you give it to her, and you give it to her. You guys are so slow. We would all be dead because we wouldn't be making enough. Now I can come over and I can give you another electron. So they have metabolic diversity that you don't have. So we can't just say that respiration is oxygen, because respiration is not just oxygen, but what oxygen and nitrate have in common, no H's. They're both inorganic. So respiration, doesn't matter where you get the electrons from, whether you're eating TSB or chicken or what, it's what are you doing with the electrons? Is oxygen taking them away? Then we call that aerobic respiration. If it's not oxygen that's taking them away, we call that anaerobic respiration. So if we look, right, here's glycolysis and the Krebs cycle, right? C6H12O6, there's the glucose. We break it into two pieces. We completely steal all the electrons from it and we give them to NADH. And NADH, that's me, right? Runs over and donates them to the electron transport chain. And at the end of the electron transport chain, here are some options. Oxygen, carbonate, nitrate, sulfate. These are all inorganic molecules that can collect those electrons. And they all become garbage because this is what you care about. So we have aerobic. This one right here is aerobic respiration because it requires oxygen. These right here are anaerobic, not oxygen requiring. So respiration doesn't mean oxygen, it means inorganic electron acceptor. And there are lots of microorganisms that can use any one of these. Right? If you got the gene to do it, do it. So E. coli, right, where you would die, E. coli would still live because E. coli could choose something else, right, and use that as its electron acceptor. So it seems like respiration is kind of the perfect plan, right? Like you've got this perfect setup, no oxygen, no problem. I'll just throw something else in there. I get a little bit of ATP here, a little bit of ATP here, lots and lots of ATP here. So yay for respiration, but here's the problem, right? So we ran out of oxygen. Well, we can also run out of nitrate, right? So we can lose the electron acceptor. If we lose the electron acceptor, we already saw that shuts down the electron transport chain. If we shut down the electron transport chain, that means that all of your NADHs are all NADHs, which means I can't go to the Krebs cycle and collect any more electrons. So if this gets shut down, then the Krebs cycle gets shut down. And if the Krebs cycle gets shut down, there's nowhere for pyruvate to go, and so glycolysis shuts down, and you die because you don't have that electron acceptor. So the result of no terminal electron acceptor is no electron transport chain, no Krebs cycle, no glycolysis, so no ATP. No ATP, you're dead. It doesn't matter if you can have 57,000 different things take the electrons. If none of those 50,000 things are available, you will die. So what do you do, right? So basically, what you're left with when all of these things shut down, you're left with just glycolysis, having produced a little bit of NADH, a little bit of ATP, and a little bit of pyruvic acid. Right? Because everything else is shut down. That's where the process stops. So we don't have any of those other things. We have NADH, a little ATP, pyruvic acid. Now, the ATP is going to quickly be used, right? So really what we have is NADH and pyruvic acid, right? Because ATP, going to use it, gone. These two, NADH and pyruvic acid, right? What are we going to do with them, right? We can't do anything with them. What we really want, what we'd really like is the electron transport chain to still work and the Krebs cycle to still work. But if we don't have that terminal electron acceptor, we're stuck with what can we do with this process right here? Because it would be nice if I could just even get those few ATP, right? So your solution is for pyruvate to get the electrons to become reduced. Pyruvate accepts the electrons. When pyruvate accepts the electrons, right? So here's pyruvate. It accepts the electrons from NADH. So that means that NAD can come back up here and glycolysis, right? So we can just eke out a little bit of ATP, right? Which you get some ATP, it's all substrate level phosphorylation, right? That pyruvate kinase and other things, right? So the main thing is that you're recycling the NAD, you're getting a little bit of ATP, but here's the problems with that solution. Number one, it's not very much ATP. Like in terms of the amount of ATP required to survive, you would die. You cannot survive only using glycolysis. It's not enough ATP for you. Now let's say that that amount of ATP is enough for you. You can grow slowly, you can take it easy, right? Your bacteria, and you're like, okay, cool. Well, here's the problem. When you undergo this process, which is called fermentation, you typically produce acids, and acids are no bueno. Or you will produce other toxic or noxious things, right? You know, when you ferment, say like beer, or wine, or kimchi, or sauerkraut, or any pickled vegetables, right? You put them in a jar, you seal it up, and they don't keep fermenting forever. You know why? Because as they ferment, all of those poisons they make kill them. Because, you know, we think of fermentation, I mean, I don't know about you, me, I love fermented things. Like I have pickles, sour, they're yummy, right? They make delicious beer, wine, right? All good, right? The reason people started fermenting things was to keep them from spoiling. Because the fermentation would make acid, and the acid would keep things from rotting your food. And that's where beer came, you know, beer, when they first came up with beer, it was actually like oatmeal. Because they didn't really want to get drunk, they wanted to have some grains that didn't get moldy. So they would ferment the grain, and they'd have this sort of soupy oatmeal, right? We throw that stuff away now and just drink the alcohol, but what they would do is cook it, and it would burn the alcohol off, and they would eat it. And then people started saying, you know, if I drink the stuff, some fun stuff happens and it tastes good. So then they started making beer and they check all that stuff, right? That's fermentation, right, was used because the result of fermentation is toxic or acidic or noxious, right? So that's why you can't hold your breath forever. Because if you undergo lactic acid fermentation for long enough, right, you go for a little bit, your legs get sore. You know why they're sore? Because there's acid in them. And it damages your muscles, and you have to work and move that lactic acid out of your muscles, right? If that lactic acid were to get into your brain, right, person who has an anoxic brain injury, a lot of times their brain will just kind of start disintegrating because all of the acidic byproducts will actually eat the brain cells. So you can end up with like a brain abscess as a result of an anoxic brain injury because fermentation produces bad things, right? But it's a solution because it still allows you to get a little bit of ATP. And if you're a single-celled microorganism, you can survive on just a little bit amount of ATP that you get for fermentation. Right now, fermentation products are often toxic, noxious. They're also delicious, right? Look at all these yummy things that we have as a result of fermentation. There are actually thousands of different things that you can eat that are fermented. Like so many, I couldn't even make a list, right? So we can't talk about all the different kinds of fermentation. What we can say is, right, there are 4 basic types of fermentation. Lactic acid fermentation, ethanol fermentation, mixed acid, and butadiol fermentation. Right? Fermentation is big time. You know, Louis Pasteur, remember, he's the guy you want to remember from chapter 1, right? Did a couple of cool things, one of which was settling the spontaneous generation debate. You know what he was studying? Fermentation. You know why? He's French. What are French people known for? Not being snooty, cheese and thing you drink with your cheese, wine. Huge industry, and their wine was going bad. And they're like, hey, Louis, figure it out. Why is my wine going bad? right? So pasteurization, we're like, yay, pasteurization makes my milk. No, it was for wine. So that wine would be delicious and they could sell it to people and not be embarrassed by their cruddy wine, right? So he was studying this, and then he heard about this whole spontaneous generation thing, and he's like, well, I know that these little invisible dudes are what's spoiling my wine, so it's probably little invisible dudes that are growing in the tube, so let's do this one neck flask thing, and Mike dropped the whole idea, right? And then biogenesis became the prevailing theory. So fermentation, right, produces lots of things that actually become useful, right? But we think about 4 things in general because that's what the bacteria we study produce: lactic acid, ethanol, mixed acid, and butadiol. So, respiration versus fermentation. Respiration has an inorganic electron acceptor. Fermentation has an organic electron acceptor. Respiration does not mean oxygen is involved, and fermentation does not mean no oxygen is involved. Respiration takes an organic molecule and completely oxidizes it to CO2. Because it has completely oxidized that molecule to CO2, it has taken all of the electrons, sent them through the electron transport chain, and it makes a ton of ATP. On the contrary, fermentation takes an organic molecule, pyruvate, and converts it to another organic molecule, say lactic acid. Look at this. This is the chemical formula for pyruvate and lactic acid. It's right, like, so there's actually more energy in that lactic acid. Not only did you not strip it completely, you actually piled more electrons on it, right? So it's like taking your bonfire and pouring water on it halfway through the bonfire. There's still a bunch of wood there that could burn, right? There's still a bunch of electrons that could be oxidized. So we have incomplete stripping of the hydrogens, which means incomplete stripping of the electrons, which means you don't make as much ATP, right? So it's not oxygen or no oxygen. It's organic in fermentation versus inorganic, right? So if we throw those 2 processes up, right? So here's respiration, right? So here is glycolysis and the Krebs cycle, this whole thing right here, right? Glucose being broken into 2, completely oxidized. NADHs go over to the electron transport chain, and an inorganic electron acceptor is used to fuel that process that makes lots of ATP. Glycolysis